Hello friends, in this video lecture I'll be talking about the restriction enzymes and the restriction enzyme modification systems. These restriction enzymes are one of the important tools in genetic engineering, and so they belong to the class of enzyme that is nuclease type of enzymes, and the role of this nuclease is to hydrolyze the phosphodiester bond present between two nucleotides. And this nuclease enzymes are of two types. The first is the exonuclease. These exonuclease enzymes coat the DNA molecule at one terminal end. That is, they coat the DNA at one terminal end. That is either from the 3' end or from the 5' end. And they remove the nucleotide one after other. So, and these exonuclease are not site specific. They are not sequence specific, and they coat the DNA molecule at one stance only. And the second, that is the endonuclease. So these enzymes coat the DNA molecule somewhere at the middle position. So means they coat within the DNA molecule. Suppose if this is the DNA molecule, so they coat somewhere at the middle. So they coat here, right? And these enzymes are sequence specific. Means they coat the DNA molecule at particular sequence, particular sites, right? So they are sequence specific, and we are having this restriction endonuclease, and this enzymes has its speciality, and the speciality that is denoted by the term restriction, right? So what is this restriction? So where this event occurs, restriction, that is, first we should know these enzymes are derived from the bacterial cells, and they are produced from the bacterial cells, and these restrictions occurs inside these bacterial cells. So now I am taking example. So I am explaining here that suppose this is one of the bacterial cell, and so this is the genome, the chromosome of this bacteria, right? And these bacterial cells are infected by one type of virus, so that is known as the bacteriophage. The bacteriophage is the virus that infects the bacteria. While it infects the bacterial cell, it delivers its gene, its genome, its DNA into this bacterial cell, right? So once inside the viral DNA in the bacterial cell, then it tries to take the control over the cellular machinery of these bacterial cells. Then the virus multiplies inside the cell or do any event. So whatever may be the event, so this becomes a danger to this bacterial cell, right? So now the bacteria tries to defend itself, tries to save from this harmful foreign genetic element, the genetic element of this virus. Now these bacterial cells have many genes, and those genes synthesize. When the genes undergoes expression, they produce particular enzymes, and they are the restriction enzymes. Only for this work, restriction enzymes, and these restriction enzymes restrict the growth of this harmful foreign genetic element. So from this event, the term restriction comes, because they restrict, they do not allow the viral DNA to make an event inside the bacterial cell, right? So I'm giving one example. One of the enzyme, restriction enzymes, that is Hy3. One of the restriction enzymes, it is named as Hy3. I will talk about the nomenclature of this enzyme later. So now this Hy3 enzyme, it is derived from the bacterium Haemophilus aegypticus, right? And this enzyme, wherever it encounters the sequence, that is 5' G, G, C, C, 3', and this is 3 prime, CCGG 5 prime, right? So wherever these enzymes encounter this sequence, then it cuts the sequence that is between the adjacent G and C, between G and C. So it makes cuts here, right? So one of the virus, that is 5X174. So it has, this virus has a circular DNA molecules. And this circular DNA molecule has this sequence at 11 places. Means this sequence occurs at 11 places in the circular DNA of this 5X174, right? So when this circular DNA of this 5X174 is treated with these enzymes, then it chops this viral DNA at 11 places, making 12 fragments. And those fragments can be separated and also can be, its sequence can also be determined, right? So in this way, the bacterial cell, the restriction enzymes of this bacterial cell code the foreign genetic element. So now a question arises. So these restriction enzymes code this viral DNA only. Why they will not code this bacterial genome? The cutting of this bacterial genome, the chances of cutting of this bacterial genome is much more larger than cutting the viral DNA, because the genome size of this bacterial cell is much more larger than this viral DNA. And this bacterial DNA have so many sites, have many recognition sequence in their DNA. So the chances is more. But such things do not happen. The bacterial cells do not allow such things to happen. So restriction enzymes cannot act upon their own DNA. So this is because, this is due to the another enzymes that is known as the modification enzyme, right? The modification enzyme means these restriction enzymes are associated with these modification enzymes. And in combination with the restriction enzymes and this modification enzymes form a restriction modification system, RM system, restriction modification system. Means the restriction modification system has two components. So first is the, so it has two components. First is the restriction enzyme, and it's the role of this restriction enzymes is to recognize the foreign genetic element, and they recognize the particular base sequence, and they cut within this particular base sequence, that is called the recognition sequence, right? And the role of this modification enzyme, the second component of the restriction modification system, that is the function of both these enzymes are somewhat same, that is the modification, their common function is to recognize the recognition sequence. The restriction enzyme recognize the specific sequence, and the modification enzymes also recognize this sequence. But instead of cleaving, this modification enzyme add one or two methyl groups to this nucleotide within the sequence, right? Means the one of the modification enzyme, that is we take it as a methyl transferase, right? So this is a modification enzymes. So this modification enzymes add methyl group to one or two bases, and this methyl group is added. Suppose I take GAATTC. Suppose it is a recognition site sequence GAATTC and CTTAAG, right? So this methyl transferase add methyl groups to one or two bases, adenine bases. The methyl group is added here, right? Methyl group. Okay. Methyl group is added. So after addition of this methyl group, and the restriction enzyme can no longer recognize this specific base sequence. So in this way, the bacterial cell, after modification of this recognition sequence, prevent its own DNA from being cut by the restriction enzymes. So they are called the restriction modification system. So now I will talk about the nomenclature of this restriction enzyme. So now I will talk about the nomenclature. So nomenclature, right? So how the restriction enzymes are named? So a restriction enzyme has four parts. The name of the restriction enzymes has four parts. So first part, second part, third part, and fourth part, right? So the first part is the name of the genus, the first letter of the genus, and it is written in capital, and also written in italics, right? And the second is the first two letter of the species name, first two letter of the species name, the organism from which it is derived or isolated. So it is written in small letter, and also in italics, right? And the third part is the name of the strain, and it is written in capital letter, a single letter, capital letter, right? Single capital letter. And the fourth part is the order of discovery, right? So I'm giving one example. That is one of the example, that is the most common restriction in the nucleus, that is echo R1. So how this naming is done, that is the E, the first letter E, that is the name of the genus Escherichia. So from Escherichia the E comes, right? So it is written in capital, and the second, this is the first part, and the second is the first two letters of the species name. So from species coli, the Escherichia coli, and the third part is the letter R. It is written in capital. This is the name of the strain. That is the name of the strain, that is RY13. From RY13, the R letter comes, right? And the fourth part is the Roman letter. So this is the order of discovery. The fourth part, it is written in Roman numerals, right? So this is one is the order of discovery. Means this is the Roman letter 1, means this is the first enzyme to be isolated from the bacterium Escherichia coli RY13, right? So in this way, the restriction enzymes are named. So in my next video, I will talk about the type of restriction enzymes and their mode of action in gene cloning.